Today we're going to talk about the Educational Particle Image Velocimetry, or ePIV system. All of the components for the ePIV system are contained within this housing. The ePIV system contains a digital camera, a laser, a lens for the laser, a variable speed water pump, a reservoir, and all of these components are aligned inside of this box and no adjustments.
Inside of the housing, we can see the laser module and the fluid reservoir.
The reservoir, located at the back of the device, has already been filled with tap water and a very small amount of polyamide C. These particles have a diameter of 50 microns and a specific gravity of 1.03. So the density of these beads is actually very close to that of water, which is
the next step is to turn on the system using the power switch located on the back of the device towards the right.
Then you're going to go to the Acquire tab on the left-hand side of the FlowX software. And here you're going to adjust the camera controls. The first of these is brightness, the second is exposure, the third is gain, and the fourth is the number of frames. These first three, brightness, exposure, and gain, all represent a percentage of the maximum range that you're using for those brightness. In this case, I found that a brightness of 55, an exposure of 65, and a gain of 75 worked well. But this might change depending on, for example, how clean the flow model is and other things. I found these numbers to vary quite a bit. In this case, I'm going to be taking 10 frames. And so once you have this set up and working well, just hit the capture button. And the software will automatically communicate with the camera, acquire 10 frames, process them, and they should show up in this window. The next step is to go to the Analyze button on the left-hand side of the page. Automatically, the software will show us two of the frames that we've captured, one right after the other. And so now we can get a sense of how this particle image velocimetry is actually going to work. If we compare one image to the next, what we can see is that clusters of particles appear to be moving in the direction of the flow. So what the software is going to do is it is going to split each image into a number of windows, the size of which is specified in pixels by this window size parameter down here. And it is going to compare each box, each window, to all of its neighboring windows to look at how particles have moved from one image to the next. The shift size parameter tells the software how far to look from one image to the next. The PIV pairs parameter tells the software how many pairs of images to compute the results for. The software will then average results over all of these individual pairs. In our case, we have 10 images, which means we have 9 successive PIV pairs. So we're going to enter 9 in this box here, and then press process. At this point, the software will compute results, and this may take a few seconds. When the data analysis is done, you'll be put back to the Analyze section on the FlowX software. Again, hit F5 to refresh the image, and now you'll see the PIV results appearing down in the two windows below. The first set shows a computed vector field overlaid on top of one of the images of the particles. The bottom image shows the computed vector field overlaid with interpolated velocity magnitudes.
By the end of this lab, you should know what particle image velocimetry is, how PIV can be used to capture images of a flow field, and how to improve the accuracy of PIV measurements.